So far in this unit, we've discussed atoms, and then ions and electrolytes, and we discussed some bigger compounds in terms of water, sodium chloride. But in this part of the unit, we're going to get into what we discussed in the first unit, macromolecules being the next level up from compounds. We'll do that by describing the structure of carbohydrates, lipids, proteins, and nucleic acids. These are the major macromolecules of the human body. How we get them, as is shown in the image here, is we actually get raw materials through the foods that we eat. The foods that we eat are made up of carbohydrates and lipids, proteins and nucleic acids. We'll talk about in the digestive system how we actually break down those raw materials, but for now we'll say we break down those raw materials into the basic building blocks of those macromolecules, and then we build up those macromolecules within our own body, within the cell. Any wastes that are produced will be discussed in the digestive system and the urinary system, but the key here is to look at this relationship, that our digestive system will take macromolecules. In this example here, we're talking about protein, and it will break down those macromolecules into smaller molecules, in this case amino acids for the protein. This is the catabolic reactions that we discussed in Unit 1. These are the reactions of the digestive system in terms of breaking down macromolecules into smaller molecules. The opposite reaction that occurs in our body then is to take those smaller molecules, like the amino acids in this image here, and build them up into larger macromolecules, the proteins that our body ultimately will use. The analogy off to the side here, those reactions, as discussed in Unit 1, are anabolic reactions, building reactions. The catabolic reactions give off energy as they proceed, and the anabolic reactions require ATP in order to occur. All of this will come up again when we get to cellular metabolism later on in the course, but it's good to start to make connections to what we're going to get into here, to what we already discussed in terms of anabolic and catabolic reactions in Unit 1. So let's start with sugar, carbohydrates. Carbohydrates are made up of carbon, hydrogen, and oxygen as the atoms that are the building blocks for carbs. In this course, we'll most often talk about glucose. Glucose is a monosaccharide of carbohydrates. Mono meaning one, and saccharide referring to the sugar. And so in this case we have glucose, which is a single sugar on its own. This is the basic building block for the carbohydrate. Another term for that is monomer. Monomer is a generic term, and as we go through these macromolecules, we're going to look at each of the monomers that build up these macromolecules. In this case, as I've said, is the monosaccharide glucose. This image of glucose here is a simplification of the glucose molecule. The glucose molecule has a chemical structure of C6H12O6, meaning that it's made up of 6 carbons, which in our molecule here would be found at each of the points of the hexagon. Then the hydrogens and oxygens would be attached to those carbons at various points around that molecule. Carbohydrates are classified according to their size and solubility. As we're saying, the monosaccharide glucose is the basic structure, the building block for carbohydrates. If we were to have 2 of those glucose bound together, we would have a disaccharide, the prefix di meaning 2. And in this case, we have 2 glucose bound together. If we were to have more than 2 glucose bound together, we would have a polysaccharide, the prefix poly meaning many. As we move along in the course, we'll see that polysaccharides are how we store our energy in our body. The main polysaccharide that we'll discuss is glycogen. It is made up of many glucose bound to each other. Next, we'll discuss proteins. Proteins can be found in all of the foods that we eat, but are usually associated with meats and legumes, the beans that are shown in this picture. Proteins contain carbon, hydrogen, oxygen, just like the carbohydrates, but then also nitrogen. If we look at the monomer for protein, it's called an amino acid. It's the basic structural unit. It's made up of a central carbon, and then bound to that central carbon is an acid group, an amine group, a hydrogen atom, and then the purple portion here in this image is one of several other molecules. Because overall there are 20 different types of amino acids that all differ from each other based on what is found in that R group area. For example, the amino acid alanine has a central carbon, the amine group off to the left, the acid group up top, to the right the single hydrogen atom, And then down below, in the R group, it has a carbon with 3 hydrogens bound to it. Anytime you were to come across an amino acid with that R group, the carbon with the 3 hydrogens, it would be an alanine amino acid. In this course, we're not going to get into individual amino acids and what their R groups are, but it's important to note that there are 20 amino acids and they differ specifically in that R group area. As was shown a couple pictures ago when we were talking about anabolism and catabolism, when we build up proteins, what's going on in the body is individual amino acids are being bound to each other, forming peptides. The bond between the individual amino acids is called a peptide bond. And as we add more and more amino acids to that structure, we form a polypeptide. So just like in the carbohydrates, we have a monomer, which is the amino acid. Then we have a polymer, which is the polypeptide, meaning made up of many peptide bonds. Next, we have nucleic acids. In this image here, it's showing DNA. However, there are 2 types of nucleic acids. DNA, which stands for deoxyribonucleic acid, because the sugar found in the structure that we'll look at in a moment is a deoxyribose sugar. We'll also talk about RNA in this course. We just won't be looking at pictures of it now. And RNA stands for ribonucleic acid. The sugar in RNA is a ribose sugar. Before moving on here, we should note that nucleic acids contain carbon, oxygen, hydrogen, and nitrogen, just like proteins. However, in addition to those 4 elements, nucleic acids also have phosphorus. You can see the phosphorus on either side of the DNA strand shown here. Specifically, the DNA has 2 strands, the one on the left and the one on the right in the expanded view of the DNA near the bottom. You can see that there's a repeating structure there of nucleotides, and if we were to zoom in on one, it looks like this. Every nucleotide is made up of a phosphate, a sugar, and then a base. The difference between the 4 types of nucleotides, the thymine, adenine, guanine, and cytosine, is that the base is different for each of them, as is shown down in the key on the right side. Our DNA contains genes, which are the blueprint for how to produce proteins and therefore directing growth and development of the body. When we get into cellular metabolism, we'll get into more detail about that, but this is the overall structure of the DNA, and then again, its role in the body in terms of providing a blueprint for how to produce proteins. Finally, let's look at lipids. Lipids are fats, and oftentimes those terms are used interchangeably. Within our body, an important fat is the triglyceride. The building blocks for a triglyceride are the glycerol, shown at the top, and then the 3 fatty acids that bind to the glycerol, forming the overall triglyceride. The term triglyceride, if we break it down, comes from tri for 3, because there's 3 fatty acids, and then glyceride for the glycerol that those 3 fatty acids are attached to. We'll get into the fatty acids a little bit more in a moment here, but the other types of fats include phospholipids. Phospholipid looks very much like a triglyceride, except for the 3rd fatty acid has been replaced by a phosphorus-containing group, a phosphate. Phospholipids will come up in our next unit about the cell when we look at the plasma membrane, the outer membrane of the cell. Finally, the 3rd type of fat that we will look at is the steroid. Specifically, this image here shows cholesterol. We'll talk a little bit more about cholesterol and its role with the cell membrane, the outer portion of the cell, in the next unit. But we'll look at it a little bit here in terms of some details about it. Before we do, though, I'll say that cholesterol is not the only steroid that we'll look at in this course either. Later on in the course, we'll talk about hormones, and several hormones within the body are also steroid-based and have a similar structure to the cholesterol shown here. Cholesterol is ingested with animal products. It plays a role in the development of arteriosclerosis, or hardening of the arteries. We won't get into diseases and so on in this course, but when you get to pathophysiology in 3rd semester, you will discuss arteriosclerosis and the fact that cholesterol is what produces that hardening of the arteries. As we said, some cholesterol is needed for structures within the body, the cell membrane as an example, and we get about 15% of our cholesterol from our diet, but 85% is converted in the liver for our own needs, produced from other lipids. Blood levels are measured in HDL, high-density lipoproteins, These lipoproteins are good for our health and collect cholesterol for breakdown in the liver. But then there's also LDL, low-density lipoproteins, that tend to deposit against the wall of the artery, leading to the arteriosclerosis that I mentioned earlier. So let's describe saturated and unsaturated fats. When we're discussing saturated and unsaturated fats, what we're discussing are the fatty acids bound to the triglyceride. Here we have a triglyceride shown where the atoms are little bubbles to give us more of a 3D image of what the triglyceride looks like. If we were to look at each of those fatty acids, they look like this. This palmitic acid is just an example of one, but what you'll notice is that each of those carbons are bound to each other via a single bond. The maximum number of hydrogens are around all of those carbons. And so overall we call this a saturated fat because it is saturated with the maximum number of hydrogen atoms that can be bound to the carbon. Compare that to this molecule. It's still a triglyceride with the glycerol on the left-hand side, and then 3 fatty acids bound to it, but then all the bends in the fatty acids is for this reason. So again, this is just an example with a linoleic acid. It's also got carbons, but you'll notice that there are double bonds between some of the carbons. That means some of the carbons only have 1 hydrogen attached to them. We call this an unsaturated fatty acid because it does not have the maximum number of hydrogen atoms that could be bound to those carbon atoms. Overall, this leads to different properties of the triglycerides that are produced here. Triglycerides with saturated fatty acids are solid at room temperature, and when we're looking at this type of fat, it's related to animal fats. This is how we as humans store our fat. Other examples include things like butter that we saw earlier. It is made up of fats that are solid at room temperature. Triglycerides, made up of unsaturated fatty acids, are liquid at room temperature. These types of fats are what we find in plants, so things like corn oil. So now that we've discussed the 4 different types of macromolecules, let's go back and look at proteins again, so that we can differentiate between essential, non-essential amino acids and explain their relationship to complete and incomplete proteins. So as we discussed previously, you find amino acids in things like meat and any food that you would eat overall, beans, vegetables, fruits. We eat that food, we digest it, and then we take those amino acids and build them up into polypeptides, into proteins. However, as we mentioned earlier, there are 20 different amino acids. They all differ from each other from the R group, the purple portion shown in the image of the amino acid. There are 8 essential amino acids and 12 non-essential amino acids of those 20. The 8 essential amino acids are amino acids that we need to get in our diet. It is essential that we take them in through our diet. When we digest protein, we break the protein down into the individual amino acids. Those amino acids cross into our blood, travel around to our body, and go to our cells so that they can build up our own proteins. Coming into the blood, the first stop is actually the liver. In the liver, there are reactions that can take place where, for the 12 non-essential amino acids, they're non-essential because we don't need to necessarily get them in our diet, the liver can produce those non-essential amino acids if needed. The liver can take other amino acids, cause a reaction that changes the R group into the appropriate R group, and we have those amino acids to go around our body. However, the 8 essential amino acids, as I stated before, we need to get them in our diet because the liver cannot convert other amino acids into those 8. We don't need to worry about which 8 are essential and which 12 are non-essential, but this is an important concept to know, especially as it relates to types of proteins, complete and incomplete. Complete proteins have all 8 essential amino acids, while the incomplete proteins do not have all 8. So where do we get these different types of proteins from? Animal sources of food have complete proteins. They have all 8 essential amino acids. Other sources of food, so plants, like vegetables and fruits and beans, are incomplete. They do not have all 8 essential amino acids. This would be important in terms of dealing with a patient who might be vegetarian or vegan. The types of food that they eat have incomplete proteins only, and therefore they need to eat a mix of different types of vegetables and plants and beans and so on in order to get all 8 of the essential amino acids and have a healthy diet. Finally, let's explain the concept of nitrogen balance. As discussed in this unit, some of the macromolecules are made up of carbon, hydrogen, and oxygen, and some, on top of the carbon, hydrogen, and oxygen, also have nitrogen. Specifically, proteins have nitrogen on top of those three other atoms. And so the concept of nitrogen balance is really getting to a balance of protein. Here's a body. Synthesis of proteins needs to be equal to our breakdown and loss of proteins. This overall is the concept of nitrogen balance, that we're making as many proteins as our body is breaking down or losing. Overall, in healthy adults, the rate of protein synthesis is equal to the rate of breakdown. However, there is positive and negative nitrogen balance, and we want to differentiate between those two. Positive nitrogen balance is when you're producing more proteins than you're losing through breakdown in the body. Some examples include pregnancy, growth of children, tissues being repaired after surgery or trauma. In each of these cases, more protein is being built in terms of growth and repair than is being lost in the body. Negative nitrogen balance includes things like emotional stress, physical stress, or lack of available dietary protein. In this case, protein breakdown exceeds the use for building proteins. These are tissues that are losing more protein than are being replaced.